Hi, my name is Rayana Anthony, and this is my project on the gut microbiome. So, to start us off, the gut microbiome is a series of bacteria, viruses, fungi, and other microscopic organisms living in the gut. It is important that certain microbes are in the gut to stabilize and nourish it. So, we compared a 7-year-old male Yorkie who had sufficient health, and he was sample 725, to a 5-year-old male pug who also had sufficient health. But this animal took heartworm supplements, and that was sample 591. So the supplements was really what we were looking at. And we chose these two samples because they were pretty similar in age and weight. We did have a closer sample, but we had to change last minute. So our hypothesis was that the microbiome that contains heartworm supplements will have a lower amount of bacteria than the microbiome with no supplements because of a disruption in the environment causing less bacteria. And ultimately we found that our hypothesis was incorrect because we found that sample 591, which was the animal who had been taking the supplements, had more bacteria, which was 3,583, as opposed to sample 725, who only had 225. So a little bit more about the gut microbiome. Microbiome is a microorganism in a particular environment. So we are specifically looking at the gut microbiome. So in our lab, we wanted to see if there would be a correlation between supplements and the amount of bacteria in the gut. A certain diet can change an individual's internal environment, but more specifically, we wanted to see if the supplements would do that too. So we also wanted to see if there would be a significant difference between the types of bacteria that were in the gut, which ultimately we did not find. So moving on to our materials and methods. For week 1, we wanted to isolate the DNA from the stool samples, and we used that with a Keigan kit. The Keigan isolates DNA from the stool and concentrates the DNA 10 times by using the Zymo ink kit. Then for week 2, we wanted to quantify and isolate the DNA from step 1 and amplify the 16S ribosomal RNA gene using the polymerase chain reaction. So we wanted to do this to copy and amplify the specific strands. And then for week 3, we used the gel electrophoresis, and we wanted to do this to check how many strands of DNA we had. So I just want to say that we ended up having to run our sample 725 first because the first read we did not get well. So that kind of took an effect on how much liquid we ended up having at the end, because we ran out of the 725 sample pretty quickly. So then we cleaned up the amplicon using Agent Court AMP XP kit, and then once the amplicon was clean, we indexed the PCR, and then we determined the nucleotide sequence on the variable 16S ribosomal RNA gene. And then lastly, for week 4, we did another PCR cleanup, and this was to remove any unused primers and buffers. And I just want to say that for the first PCR cleanup, that was also to remove any unused primers or buffers. But moving on, then we used the fluorometric DNA quantitation assay, and that was to determine the concentration of small amounts of liquid. And the reason we use this instead of the spectrophotometer was because this is more sensitive. Then we determine the molar concentration of the index PCR, and then prepare the dilution of DNA for sequencing. And then we did the DNA sequencing again through the DNA analyzer. And then here's a picture of our gel electrophoresis. You can't really see it, but this is sample 591, which is really clear, but you can barely even see sample 725. And this was our second time running it, so our first time running it, you could not see it at all. All right, and then on to our graphs. So this right here is our positive control, and this is our negative control genus. And I chose these two specific classifications because these are the most specific classifications you can get. So for our positive control, you can see that there were 6,859 Escherichia strands of bacteria, and that makes sense because that's a strand of E. coli. And in total, there was 8,503 strands of DNA for the positive control. And then moving on to our negative control, in total there was 168 strands, strands of DNA, but right here, and right here what it's doing is classifying the bacteria. You can see that at the genus level, the first line, it says unclassified genus level, and that makes sense because we were just simply using water. Okay, and then moving on, this is our kingdom level for sample 591, which was the sample that took supplements. This is our order, and this is our genus. So I just want to start off with the kingdom. You can see that we got 3,581 reads, and it was found that 99.94% of our sample was bacteria, which is what it should have been around. And then at the order level, there was 1,407 strands of the bacteriodales, and then at the genus level, there was 1,366 strands of bacteroids. And this is actually really good for what we found. Well, good for the dog, but it disproved our hypothesis because this same strand of DNA was actually found also in our genus for sample 725. You can see there's 107 strands of the bacteroids, and the bacteroid strand of bacteria is actually just a strand of bacteria that should be found in the gut microbiome. So they both have the same bacteria, and it's also healthy bacteria, so the supplements did not cause a significant difference in what kind of bacteria would be found. All right, but moving back over here, this is our kingdom for sample 725. This is the sample without the supplements. You can see that 227 strands of DNA were found. And this is our order level. So once again, you can see that at the kingdom level, there for sample 591 with the supplements, 3,581 strands of bacteria were found. And at the kingdom level for sample 725, which had no supplements, only 227 samples were found. So once again, our hypothesis was disproven because the sample with the supplements has way more bacteria in it. All right, and then moving on to the discussion. So in extracting DNA from sample 591, we found that we got a good reading on the amount of DNA present. However, for sample 725, we found that we were not able to get a good reading of the DNA as we would have wanted to. We got a high concentration for sample 591, so this means that there was a high amount of bacteria present. So there, once again, there was the 3,583 bacteria in total. And then for our sample 725, we only had the 227 number of reads. So that was all the bacteria that had passed through all the quality sample filtering. So due to there not being a significant amount of bacteria present, we feel as though we do not have enough to base our analysis off of. So for both samples, we found that the genus Bacteroides was most common in both samples, and in sample 591 there was a reading of 38.12%, while in sample 725 there's a reading of 47.14%. And according to the Science Direct, Bacteroides is a genus of gram-negative, obligate, anaerobic, non-sporulating bacilli that belongs to the family Bacteroideaceae. So they live within the lower intestinal tract and are conventional bacteria. So once again, that type of bacteria that was found in our two samples should have been there. Alrighty, and then in conclusion, our hypothesis was incorrect. We stated that we believe there would be less bacteria in sample 591 because of the supplements that the organism was taking. However, more bacteria was found in sample 591 than in sample 725. And here's our references.